I guess background is sort of interesting. So Jochen invited me in 2019 to Berlin. I was really nervous to come over. I was the only biologist at this last workshop, and it's just been really nice to interact with everyone who has different backgrounds, but this shared interest in dormancy and latency. So yeah, I'm learning a lot. It's nice to be exposed to equations and theorems and proofs, and a second time after reading things. And I guess the other thing that came out of all this is when I came back from Berlin in 2019, right before the pandemic, we started having our Monday night sessions with Jochen and Maita and Frank, and we did it almost every week. And it was a lot of fun, and we started to get some of our ideas together. We started to agree on common language, and I think appreciation for what each other does. So anyways, it's great to be back here and think about some of these things with you. And this is one of the figures that a slight modification of the figure that we've seen variations of over the course of the week. And so just a few review some of the basic properties of this model. First, there's two pools of individuals, what we call N and M in these coalescent models, the number of active individuals and the number of inactive individuals. And then those individuals can move between those two states via these transitions in dormancy, which can be responsive or they can be stochastic. And not only can you encode those mathematically, but in the real world, that happens. We have evidence in cases where there's definitely responsive switching, but also stochastic switching. The differences in mortality rates are important. We've talked a little bit about that, but in general, we assume that mortality is higher for individuals in the active state, and that the benefit that individuals enjoy in dormancy is that they have reduced rates of mortality. That doesn't last forever, though. There's usually some constraint on how long an individual can persist in that state, just due to a whole suite of biological factors, at least from my perspective. Then we've talked a lot about Matthias and Daria. We've talked and Nathan about this idea that dormancy and which is dispersal in time is not independent of dispersal in space. And those two things can interact with one another. So it's nice to see that the group as a whole in the past few years is starting to think about the interplay between dispersal and dormancy, both in an ecological and a genetic context. And so the last thing I want to point out is the colors that are contained within these two pools of individuals. So in the coalescent sense, we could think about big A's and little a's. And we can think about mutations, some of which may be neutral, but a lot of them will have fitness consequences. They could be either deleterious for the fitness of an individual, or they could be beneficial. And when they reside in that seed bank, that can have consequences. We could imagine that a plant seed that germinates in a dark forest, if it has a trait that allows it to be tolerant of shade, then that would be beneficial and could allow for the persistence of a plant species. Or in the context of viruses, I'm going to show you, we have some data that certain phenotypic traits and their underlying genotypes are preserved in the seed bank, and specifically with regard to virus resistance. And then I guess we all should consider that a lot of those colors, the motivation for all of this work came from some the techniques that Nathan showed, where we could take a liter of water or a gram of soil and use microbiological techniques where we extract nucleic acids, which is a common way of characterizing who's there. But when we looked at who was there and compared it to like what was active, these communities were completely distinct. And those were the initial motivations that led to this work, is that it was insufficient to just know who was there and to understand biological patterns, dynamics, and phenomena. So yeah, so dormancy generates this seed bank, and there's a lot of complexity that can emerge from that that structure. And so in this paper with Frank and Jochen and Maita, we identified what we meant by complexity, and that there's emergence behavior from these individuals and their decisions to go back and forth in between the dormant states. We know that these things are operating across scales of both time and space, which could be from, microns to meters to kilometers, from minutes, hours to millennia. So that's a complex feature of seed banks. Obviously, this is creating structure, which I've already described. In parts, memory, we've been talking about delays this week, and also the potential for that to feed back on the system. So I think a lot about dormancy, but I don't expect everyone to. And maybe there are some situations where you don't need to worry about this. But I think there are probably instances where in biology, where there are dynamics or patterns that can't be accounted for with traditional theory that doesn't consider dormancy. So that's how I tend to think about this. And so I think I was going to do one thing, but then I was encouraged to maybe do two things. And I'm wrapping that up under this umbrella of complexity. And so briefly, I'm going to show some simulations that a student of mine has been working at, where we're thinking about complexity that emerges within a population using some really simple rules. And then I'm going to think about complexity in the context of the spirit of the workshop, which is thinking about viruses and how they interact with hosts and how dormancy can modify the outcomes that we might expect. So yeah, so this is what Simone and I were talking about the other night at the picnic. This work that a student of mine, Pat, is working on. And Pat is a PhD student in informatics and is interested in complex systems. And over the course of the pandemic, we started talking about these models that were developed in the 1970s by John Conway, a mathematician, called the game of life. Has anyone heard of the game of? Okay, I'm seeing some nods, so that's good. I feel like I was a little bit unsure whether or not I should be describing these simulations to a group of mathematicians. But so imagine that there's a two-dimensional grid made up of cells. In this case, the grid is like maybe 40 by 40. And there's each cell can have one of two states. You can either have a live individual that in this case is represented by blue, or it will be dead or unoccupied and white. And so then the rules are applied to a local neighborhood consisting of this 3 by 3 grid that's expanded in the middle. And you focus in on the center individual. And then we apply these simple rules. So this is vanilla version 1970 game of life. And so rule number 1, if you are a live cell in blue and you have less than 2 living neighbors, either in the vertical, horizontal, or diagonal direction, then you die. And the reason, the biological explanation would be, is that you have too few individuals. So maybe there might be some allele effect, or you just need neighbors to reproduce with. So that's underpopulation, if you will. If that living cell has 2 to 3 neighbors, then that means the conditions are good. And in the next time step, that individual would remain alive. If you have more than 3 individuals, now you're overcrowded, and so you will die. And if you're a dead cell, white in the center, and you're encountered by 3 neighboring cells, then that's a signature for reproduction. Okay, so that's the game of life. And people have used these and variations of these rules to do all sorts of things, explore chaos. These are all deterministic, right? So there's many ways you could start one of these configurations is the term that we're using. And so I'll explore that in a little bit. But the rules, if you run these, the same initial configuration, you get the same outcome every single time. So we started thinking about dormant life. And if you could take one of these Conwaying type models and introduce dormancy. And it turns out there's actually somebody who did this in 2006, but it wasn't very well developed. It was published in a book and it hasn't really received a lot of attention, and the rules were a little bit weird. So we've been playing around with a little bit of updating, still trying to keep things very simple, but introducing some variations which we think might capture the essence of dormancy. So first thing we need to do is introduce a new state. So there's now 3 states. There's live, dormant, and dead. So now if a live cell in the center, in this case, orange, has no living neighbors, it's underpopulated and it will die in the next time step. A live cell with 1 living neighbor will go into a dormant state instead of dying. Same rule number 3 is the same. If you have live cells with 2 to 3 living neighbors, then you live in the next time step. Number 4, if you're a live cell and you have 3 or more neighbors, again, you die. And the 5th rule relates to resuscitation. If you're a dormant cell with 2 to 3 living neighbors, then you will reactivate and transition back into the active pool. So we could argue about whether or not these are reasonable rules or not, and we're exploring like the sensitivity of the outcomes of these models to some of those rules, but that's what we've been doing. And it's very initial. Everything I'm going to talk about today is unpublished and still kind of in progress. So if anyone has any thoughts or feedbacks, that would be fine to interrupt. So I'm going to show you a movie now. And before I start the movie, what I'm plotting, and those lines will pop up, is the fraction of living cells. So either dark blue or orange through time. Yeah. So they both started about 25%. It's like hiccuping a little bit, so hopefully we'll get through the movie. But you can see that without dormancy, the traditional version 1.0 game of life, that the fraction of living cells goes down pretty quickly. And there's just these 2 objects or formations that you see that are called still lifes. There's nothing interesting going on. They're still updating. The rules are still being applied every generation, but it's not going anywhere. It's reached a dead end, if you will. But with dormancy, you can see that there's some bouncing around, but it's pretty persistent. Looks like it's some kind of equilibrium of about 25%. I thought this was a little interesting because I didn't necessarily, it seemed sort of counterintuitive that you could allow for physical space in these simulations that would be made up of dormant cells. So 1 out of, just by chance, 3 positions would not be, 2 out of 3 would not be living. Yet we see much more living cells maintained through time. So this is just one example simulation from what we've been doing, Pat and I. Okay, so the next thing, this is like last week, we started playing around. This is the great part about going to meetings, right? You're forced to kind of like sit down and think about your work and bring it together. I'm going to show you are some configuration maps. Because these are deterministic, we can take a small 3x3 grid and look at all the possible initial states and their outcomes, and look at them in a network or as a graph. And they look, as maybe you would expect, really different. So on the left-hand side, this is sort of a zoomed out version of the entire configuration map. So every single node in this network represents an initial starting condition. And then you can follow that to its next updated time step, the next updated time step. And there's a couple, and then on the right, you're looking at a zoomed in square region of this game of life configuration. So there's 2 states, live or dead, 9 possibilities. So 2 to the power 9, there's 512 total starting configurations to consider. Yeah, and so what are the observations? I think that there seems to be somewhat modular. There's little distinct groupings of starting conditions, and in this one particular large group, all of these are configurations that are 1 step away from extinction, marked in blue. And there's other ones that are similar to this. So when we look at the dormant life, now there are 3 states raised to the power of 9. So there's roughly 20,000 configurations that we need to consider. And there's a much different topology of this configuration network, if you will. And they're different. I mean, you still see some of these groups, these modules in space, but these lines, as you might be able to see, means that there's multiple steps that you have to go through to get to your final state, which could either be one of those still lifes or extinction. So in addition to seeing a higher percentage of life being sustained, which I think is generally consistent with the idea or the notion of a seed bank, that it prevents, that it can sustain populations through time and aid in persistence. Another intuitive feature from these figures is that it seems like it's harder for the system to go to extinction when you include this third dormant state. So, you know, I don't know how biological this is. I don't know how realistic the rules are. I'm sure there's better mathematical ways to do something like this. So I was like really embarrassed to even show it because it's new. And I know there's people who are thinking about interacting particle systems, and there's probably much better, more sophisticated ways to think about this. But this is sort of like a pandemic toy that I've been playing around with, Pat, and I'm sure it'll get more sophisticated with time. Okay, I'm going to move to the second part, which is now viruses but putting this in the context of complexity due to species interaction, which is something that Nathan raised. So this is just an example of different types of modules or food web configurations that biologists sometimes think about, and they can vary in their connectance and the number of nodes that you have. And the reason why I'm showing this is because we often think of dormancy as being something that's favored by extrinsic environmental forces, starvation, high elevated temperatures, pH, et cetera. But a lot of noise and fluctuations can be generated endogenously through species interactions. And we started to wonder in the process of writing this paper and some work that the ideas when Nathan was in our group, that maybe those endogenously produced fluctuations could reinforce or modify the conditions under which we might see seed banks emerge. So that's the general idea. So this is how we're thinking about host-parasite dynamics with a seed bank. So I won't describe the dormancy module on the right because we've all seen this a lot, but we consider resources that are consumed by a host population. And that host population has heterogeneity in the sense that individuals can go back and forth between active and dormant states via all the rules that we've discussed. But then the host is interacting with a parasite population P. So this is the conceptual way we're thinking about the integration of dormancy in a host parasite food web. R is the resources that are being consumed by the host. Okay. I'm going to move on now and talk about a little bit more biology. So in our group, we've been working with bacteria, and we work with a group of bacteria called Bacillus, which is closely related to Clostridia, which are bacteria that live in all of our guts. And they have this unique ability to form these structures called endospores when the cells are starved for things like amino acids. The ability to do this is really special, and it's really old. People have estimated that perhaps the last universal common bacteria on Earth was a spore former, which dates it somewhere close to 2.7 to 3 billion years ago. It's also a very complex trait. To make an endospore requires something like 500 genes. In some cases, up to 10% of the entire genome is dedicated to making these resting structures. And I'm not going to go through this diagram here, but you can see in the blue the good conditions. When a cell is happy, it gets bigger, it divides. It gets bigger, it divides. So it's just binary fission. But when it's induced, so this is responsive induction and waking up in most cases, it enters this really complicated program that involves expression of these special accessory genes complete modification of the cell, genome duplication, and the production of this resting cell at the end. The spore in the end is one of the most biologically inert substances on the planet. The spores can sit around for millions of years. You can resurrect them from halite crystals, amber, permafrost samples everywhere. And in fact, some estimates by some German researchers suggest that there are just in the ocean sediments alone, there are something like 10 to the power 29 spores on Earth. So they're everywhere. The Earth is littered with spores. And on the left, I guess, is a little GIF that's basically showing a picture of spores underneath a microscope as they wake up in response to resources being supplied. So you're witnessing responsive resuscitation in these cells. What is the cost of making a spore? I think Felix alluded to this. Jerry Cascella, who's Warwick, he used this language when we were talking in 2019. And we were just talking about this outside. The notion, he said, well, sometimes when dormant individuals fall into dormancy, and it was this envision like this passive sort of process where maybe if you don't have enough nutrition, enough energy, you just kind of as a default become dormant. And that's not the case with the formation of endospores. First, there's opportunity cost. So to undergo binary fission, which I described, takes on average 20 minutes. To make a spore, it takes 25 times longer. It's 8 hours to make a spore. There are some interesting stages in between called a commitment stage. Like you can go up to a certain point and you can change your mind and go back to a vegetative cell because a lot could happen in 8 hours if you're dividing every 20 minutes. But once you pass that point of commitment, that checkpoint, you can no longer go back to being a vegetative cell. So it's a long journey to make a spore. And a lot can happen. A vegetative cell during 8 hours could make 10 million babies. So you have to be careful about the decision of making a spore. So that's opportunity cost. But we were interested in the bioenergetic cost. How much ATP, the common biological currency for all life, how many ATPs does it cost? And if we had that information, we might be able to relativize it to other biological processes or incorporate this into models. Because one of the things that's interesting is that despite being around Earth for 3 billion years, if we grow these cells under good conditions and maintain them under good conditions for a couple weeks, you relax selection on major portions of the genome, and random mutations will eventually hit one of those essential genes that's required for making a spore. And you can see this ancient complex process just crumble and fall away, right? So, Felix, your logic is, well, this must be really advantageous because it's costly. But because it's costly, it can be purged from these populations pretty readily as well. So Janin Karkoch, she was a PhD student in Germany. She got her degree in Leipzig, came into my lab last year and started trying to use these techniques that take into account the amount of energy that it takes to copy a gene, transcribe a gene, and translate a gene. There's 500 of them, remember, some of them vary in length. But the major cost is translation, making proteins out of those genes. And so I've highlighted the cost as sporulation, and on the upper y-axis, just rescaled that to 1. So we have like 100% of the cost to make a spore. Maybe that the units of ATP maybe aren't all that necessary, but you can think about the relative costs related to other things. So homeostasis, just maintaining basic metabolism, basal metabolism and homeostasis, it's like 200 to 300 fold more expensive to make a spore than just to hang out and do nothing. And things like maintaining the essential genome, all the genes that are needed for life, it's something like 40% of the cost of that. And it's somewhere about 3 to 4% to just compared to the ability just to divide, to undergo reproduction of binary fission. So again, these are sort of new data that we've been crunching and trying to interpret as well. But I guess it matches our intuition based just on the time and the opportunity cost that from a bioenergetic standpoint as well, this is a really costly biological process. Okay. But these costs are just on a per cell basis. We've talked a little bit about bet hedging this week. So we might ask ourselves how many, under a given set of environmental conditions, either at one point in time or averaged over time, how many cells should undergo this process? Is one sufficient for maintaining the population, or maybe you need more? These populations in a test tube can easily reach, 1 million, 10 million per milliliter. So how many of those cells should undergo sporulation given the costs on a per cell basis? We don't know the answer to that. Maybe we could model that and think about optimality. But what we thought at first we would do, often what we do now, is just go to the literature before we start lifting a pipette or doing anything, and just seeing how much variation is there in the sporulation efficiency, which is the fraction of cells under a given set of conditions that will engage in this process, that make the decision to go dormant. And these are the data. On the right is a picture that we took in the laboratory with a new microscope, which I think I didn't take it, obviously, but I think it looks beautiful. And you can see that there are some vegetative cells in black that don't have any of those white objects. And the white objects are the endospores that form within the mother cells. So there's some of the black cells that don't have spores, and there's many that do. So sporulation efficiency ranges from 0 to 95% of the individuals under varying sets of conditions. And the central tendency is at about 30% on average. So we can take those costs on a per cell basis and start to multiply them through the number of individuals in a population. And it seems like that would be costly. Okay, so that's the background on the host. And what I'm going to develop now is the interactions that occur between these spore-forming bacteria and the viruses or phage that infect them. For those who aren't familiar with how difficult it is for bacteria to make a living, not only are they starving for resources from the bottom up, but they got to deal with a lot of infectious phage particles. So for every single, as a rule of thumb, for every single bacterial host in your gut or in the ocean or in the soil, there are about 10 virus particles that are floating around randomly in the environment via Brownian motion, waiting to just potentially attach and irreversibly infect, adsorb to the cell and cause an infection. So how do we study this, at least for the purposes of the talk today? There's these in the middle. What I'm showing you is a Petri dish, and you can see the small little circles there. Those are plaques. When Lior was here, he was talking about plaque forming units as a way to enumerate the abundance of herpes viruses in a sample. So it's a similar approach here. So there's the gray here is a lawn of bacteria of the bacillus that's growing up underneath a firmer layer of resources. So the lawn, imagine like a grass lawn, is growing up off of the resources. And then there's a dilution of viruses that are put in there. So you can account for the dilution. 2 days later, you see those plaques form, and you can count them, and you can say, there was 10 to the 7th virus particles in this sample. There's some other information that we're going to extract from this type of assay as well. So that's one way to study viruses in the laboratory. But we often aren't able to culture all of the viruses that are out there. So another technique that's emerged over the last 10 years or so is to characterize viruses using genomics. In this case, you just take all of the DNA and you sequence it and you look at the genes. And people are really interested in describing viral communities in this way. I'm going to show you hopefully some evidence to suggest that using genomics and culture techniques that viruses can overcome host defenses of dormancy by stealing dormancy genes and manipulating their hosts, so hijacking the dormancy metabolic pathways. So just quickly, here are the ways that I'm conceptualizing interactions between bacteria and viruses and dormancy. So first, on the left, we consider that there's just no dormancy. How do these viruses? They don't do any of the stuff, interesting stuff that herpes viruses does or HIV does. These are just little machines that, again, are floating around. They attach to receptors. They inject their DNA. They make new virus particles, and then they produce an enzyme that lyses the cell, and they're going to release somewhere on the order of 50 to 200 virus particles into the environment, which are just going to float around and look for new hosts. So really, from an evolutionary perspective, though, is a really strong selective pressure on the host populations. And we often see a rapid evolutionary change occurring due to this pressure of virus infection. So what does dormancy do to modify this basic lytic infection cycle? Well, spores can't be infected. This is the initial first stage of contact. This is called an adsorption assay. So we take some cells and we add some virus to them. And then over time, we just measure the concentration of free virus particles, because again, these viruses are just moving around randomly, encountering hosts and irreversibly absorbing to receptors. And so in the green line with the vegetative cells, you can see that there's a logarithmic decline in the concentration of free virus particles over time. And that's just because they're attaching to hosts. But in the orange line, which is the spore fraction, you can see that the percentage of free virus particles is at 100%. It's not changing over time. And it's because that spore has been physically, morphologically remodeled in a way that there's no way for it to effectively attach and infect. So this is what we kind of are thinking of as either resistance or a refuge, a dormancy refuge from virus infection. Maybe this balances out the cost, right? It's not just surviving through time, but you're getting this added benefit of not dying from being infected by these viruses that are in your environment. I'm curious if, do viruses have anything to say about this? Is this can they get around this in any way? I alluded to I gave you some heads up that we're going to revisit this with genomics, but there are some other ways that viruses appear to have been able to get around this. And it has to do with the timing of the development of the spore relative to the time it takes to infect. Look at the time scale in the second panel. We're talking about 0 to 10 minutes for infection to take place. But remember, it takes 8 hours to make a spore. So you can experience bad conditions in your environment, and you say, all right, we're going to start the sporulation process, but you're still susceptible to all these viruses that are living in your environment. So those viruses can enter the vegetative cell, and their genomes become aligned with the host genome in the development of the spore, and they get physically translocated from the mother cell into the daughter cell. And what results from this is what's called a virospore. The naked genome of the virus is just hanging out, unincorporated in the host genome, taking advantage of the refuge of the spore. So when your environment is not good, the virus genome can just hang out inside of this structure and be protected for however long until good conditions resume. That's when the virus can start its infection cycle again. So it favors survivorship component of the virus as opposed to reproductive fitness components. That happens. I'm not going to show any data on it, but it suggests that it's not things that I'm just like making up, or that's a convenient way of testing ideas and theory. That there does actually, as I study this more, there are observations that we're making both in the laboratory and in the literature to suggest that this is a real thing, that there could be some co-evolution going on, and that viruses are trying to get around this dormancy defense mechanism. Okay, I'm going to talk about a project that came about recently when we talked about the touch of dormancy. Andras had his talk where viruses can contact bacteria and that can initiate transitions into dormancy. That work was done by Joshua Weitz. And so he's been collaborating with us. He's a mathematician and physicist, I guess. And one of his students wanted to come into our lab and get some wet lab experience. So here's an example where we have collaborations ongoing between mathematicians and biologists to explore some questions. And so I'll show you, the student showed up and we had to think about what we could do as a project to give her some experience. And it turned out to be sort of interesting. And this is very much still in progress as well. I talked about plaques, right? And mostly we use these just for enumerating the abundance of viruses in a sample. That's pretty straightforward. But I've always been curious about what limits the size of a plaque on a lawn. So remember, there's resources in the media below, and then we plate the cells and it creates this lawn. And then each one of those plaques is formed from a single viral particle that grows up, replicates, and expands. But it can only get so big. And the observation that we made is that we used different types of bacillus hosts. So another great feature of bacillus is that it's a good genetic system, meaning that you can manipulate and do genetics and knock out certain genes, including ones that are essential for forming spores. And so that's what we did. We removed a gene that was critical and essential for the formation of spores, but was otherwise not costly. And then we infected these cells and grew them on Petri dishes and looked at the morphology of the plaques when the host could engage in dormancy and when it couldn't. And so the orange lines here, what we're seeing is the plaques. These are made from lots and lots of movies that Andrea Magali, the student at Georgia Tech, did. And so she measured in movies the size of multiple plaques over time in the spore forming host, and they're about 25 millimeters in size. But when we knocked out a gene that was required for forming spores, you have non-spore forming hosts, you can see that the growth rate measured by plaque radius over time is much different. And in the end are about 5 times larger than the hosts that engage in dormancy. So that's an interesting pattern, and it's consistent with the idea that maybe sporulation prevents the spreading of viral epidemics in this 2-dimensional plates. Okay, so then we're like, well, maybe we need to take a little bit closer. This is the plaques are sort of a macroscopic view of what's going on. You can measure them with a ruler, you could take pictures with your iPhone and get a sense. And we thought maybe we would devise a way to put these, make little mini plaques on Petri dishes and look at them under the microscope, because we have these techniques for distinguishing between spores and vegetative cells. And so that's what we did. We grew up these little plaques on microscope plates, and the clearing in the middle, all the dark area, is where all the cells have been killed by viruses. And around the perimeter, the leading edge of these plaques, we see an enrichment of spores. So it sort of seems, or one could hypothesize, that maybe spores are preventing physically the spread or diffusion of viruses and limiting the size of the plaque. What's in the center? This is, we're kind of zoomed in on a plaque here, and all of these cells, you don't see there are a few cells in there. I'm not going to get into that, could be resistant evolutionary mutant bacteria that are evolving, but this whole area is like a desert that's free of any hosts. And these cells around the perimeter are all spore forming bacteria. It is why there's all of the viruses. We can't see the viruses here. I should have pointed that. Yeah. But this is the close-up vision of what's going on here. And the major point is that when you look around the edges of these plaques, which on average are 5 times smaller than the ones that can't form spores, there is an enrichment or wall of spores surrounding the edge, and that suggests that maybe that's contributing to the diminished spread of the viruses on this 2-dimensional availability of hosts. Does that make sense? Okay. So we thought Andrea, Joshua, Daniel, as a postdoc of mine, I thought we would try to explore this using some partial differential equations. I don't think there's anything that's like super hard to understand here. The one thing that we make assumptions about are that the resources and the viruses can diffuse in the plate, but the bacteria are the lawn is fixed in place. And so resources are depleted through cell growth. Resources are changing over time due to diffusion from neighboring cells in this lattice, which represents the Petri dish in XY coordinates. They grow according to some separate function where cells increase as the availability of resources there. They die from infection and absorption rate. And then there's a resource dependent function that controls the transition of active cells into dormant cells. We don't have a resuscitation term. These are relatively short-term experiments and there's no resupply of resources. So maybe we should include that, but for first pass, we decided that the only transition is active cells going into dormant cells. There are infected cells, so active cells come into contact with the virus particles, they become infected and they die according to this function eta. Some of those virus particles can just decay, which is normal if they don't get inside of a host. And then the viruses are just dying from lysis, decay, infection, and then there's some positive gains spatially due to diffusion again. So that's the underlying set of equations that Andrea had developed. And there's some movies that hopefully I can show. So this is the 2x2 grid. We're just looking at the total bacteria. So the sum of, because this is what we see when we grow them up on plates. So it's the sum of spores and dormant cells and active cells. And blue is low abundances and yellow is going to be when there's a really thick lawn. And there's one virus that's introduced in the center of this grid. So there's the lawn that forms in yellow, and then you can see the spore forming in the center, growing. This is again without dormancy, right? So there's nothing, that rampart of spores isn't there. They can just keep on growing, and that's about what they look like at the end. And then if we do this with dormancy in the model, you can see that they, on average, are in the experiments, maybe like overnight. So there's a 20% difference. I mean, there's a lot more to be done in terms of exploring parameter ranges, et cetera, and probably other. It's what I can tell you experimentally is that we thought about that maybe there's like when cells lyse, maybe they release chemicals into the environment, which accelerate spore formation. We've done some experiments on that and we don't see that that's the case. So cells aren't able to detect the guts of their neighbors and say, oh boy, we better go dormant. That doesn't seem to be what's going on. What we think is going on is that there's something about the rates of resource depletion and how that influences the timing of sporulation. And so this may get a little bit geeky, but we've been able to get new mutant strains that can delay the onset or accelerate the onset of sporulation. And we think that might be a good test for that hypothesis. The point is, it seems like when we're looking at, this is one demonstration that sporulation seems to inhibit the spread of viruses in these populations. Okay, so now I'm going to show you another set of experiments. These are co-evolution experiments. And so this is something that ecologists and evolutionary biologists are really fascinated by. And there's not really always really great examples that there's reciprocal evolution, that a change in a gene in one population will lead to a corresponding change in another population, and that you can generate this evolutionary feedback. Perhaps for 20, 30 years, maybe more, people have used host phage systems to study as a model system co-evolution. We know a lot about the arms race dynamics and the directional selection that occurs when you add a virus to a host population. And so our question was, all right, we'll take that as a given, and we want to ask whether or not dormancy changes the co-evolutionary outcomes that we would otherwise anticipate. So quickly, I'm going to go through, I figure maybe not everyone is really familiar with the notion of experimental evolution, but for a long time, biologists would just observe evolution in the field. And with microorganisms in particular, but other groups of taxa, we can now study this in the laboratory. And microorganisms are a really great way to do this because they're really large population sizes, fast growth rates. And one of the things that you can do is you can transfer them from time to time to time and let them go for prolonged periods of time, either in serial transfers, like I'm going to be talking about today, or in these devices called chemostats. And over time, you can take individuals out and put them in the freezer and create effectively a fossil record, which allows you to later on, for example, do these competition experiments that we've heard a little bit about this week and find out whether or not certain lineages are more fit than they were early on in the experiment. But we can also sequence their genomes and determine what types of new mutations are arising under a given set of conditions, how many, what's the rate at which they fix into a population, what is the nature of the putative function, are those neutral or adaptive mutations? So experimental evolution as a method allows us to address a lot of those questions, but I don't think it's been necessarily used to investigate questions pertaining to dormancy. And so that was part of what we were wanting to do here. In the lower right, this is just a picture of this phage. This is where all the DNA is held. And these are the tail fibers that attach to the receptors on the bacterial cell. And these are the little flasks. They're like maybe 10, 15 mils of media with cells, and we put them in an incubating shaker at 37 degrees or whatever, and let them grow. And every day or so, we'll just take a fraction, 10% usually, and put that into fresh media. And we just propagate that and do that over time. In this experiment, I think we did it for a month. So I want to just take a moment, though, to talk to you about this treatment that we generated, which is a seed bank treatment. And so if we don't want dormancy to be a factor in some of these vials and these replicate populations, we use one of those genetic mutants where we deleted one of the genes that's required for sporulation. So in these populations, they get transferred every day or every other day, and the bacteria that are growing up and then are gray are obligately vegetative. They can't engage in dormancy as a defense against viruses, which we're going to introduce. The seed bank treatment is a little bit different. First, we're starting with the wild type host, which can form spores, but we created an external seed bank to try to enrich the number and the history of lineages that would be in these populations. So over a moving window of 3 days, we would take a subset of these cells here, put them in little tubes and heat them at 80 degrees centigrade for 10 minutes. And that kills off all the vegetative cells. But remember, I told you that these spores are really resilient. So we can enrich for the spore fraction, combine them over a 3-day window, and then add them back at each transfer over time. So we're not just relying on its capacity to form spores, we're externally enriching for a history of cells from previous time points. If you don't follow that, it's okay. We have a seed bank, we have a no seed bank treatment, and we're gonna let them run for a while, and we're gonna measure a few things. The first thing we measured was population dynamics. Joachim was telling us about how the seed bank can affect population dynamics. And so here we're just measuring the abundance or concentration of cells in those replicate populations over time. The phage population actually was not, here in black, was not really affected by the seed bank, but you can see that there's about a 2 order of magnitude drop in host densities in the first 2 weeks of this experiment, which is pretty significant both biologically and statistically. So the effect, the seed bank affects host population densities and dynamics over time. And so what I mentioned is we can do this, we can create this, we can take individual cells out and we can freeze them down and grow them back up. And one of the things that we can do is we can, for each time point, we took out 20 colonies and we challenged them against the ancestral virus, the virus that we started the experiment out. And what I told you is that the rapid evolution is the norm. So if you add a virus and you come back a day later, most of those individuals, the selection pressure is so strong that there will no longer be any susceptible cells. And this is what happens when you have directional selection. You're getting replacement of phenotypes over time. More and more selection for more and more resistant phenotypes. But you're also purging genetic and phenotypic variation in that process. So this is the selective sweep that we've talked a little about this week. And the question is, would the seed bank potentially slow down the rate of those sweeps by preserving genetic variation in the spores? Okay, that's the motivation. And so if we calculate the fraction of those 20 cells that were taken from each tube, so a total of maybe 50 to 100 of them per time step, and we calculate the fraction of those that are resistant to the ancestral phage, it's very low. So almost 100% of these cells are now resistant after introduction of the initial virus population. And that's true in the seed bank fraction too, but only for the vegetative cells. If we look at the spores and we hatch those open and grow those bacteria, you can see that the maintenance of the sensitive ancestral phenotype, they retain their sensitivity to the initial phage population. So that's cool. I'm not sure if I articulated that well, but I think that's a basic assumption of what we should expect from theory in a seed bank is the maintenance of genetic and phenotypic variation that would otherwise be purged from the population. Okay, last thing that I have for this experiment, we can sequence the genomes of all of these bacteria. We're now getting to a point where that's cost effective, easy, and pretty high throughput. So we have the genome of the ancestor, and then over time, we sequence the genomes of the bacteria and those populations that have had different experiences, whether it be the seed bank or viruses. We map those sequences back onto the ancestor and we look for any changes that occurred. And those are mutations, single nucleotide mutations. And so we did that and we built a mutation by sample matrix. And what I'm going to show you is a simple dimension reduction analysis, a principal coordinates analysis. It's going to show the composition of mutations in those different populations that correspond to those different treatments, the presence or absence of the virus and the presence or absence of the seed bank. Yeah, okay. And here it is. So the first axis here is explaining 50% of the variation. And this you can see is due to the seed bank effect. So samples over here are going to be associated positively with the seed bank. And the ones over here are going to be positively associated with, come from populations that are not in the seed bank. And on this axis here, PCOA2, you're looking at the phage effect. So not surprisingly, you add a virus and these populations start to rapidly undergo evolution. They're acquiring new mutations. And that's why you see this big separation from these populations and these populations. Lots of mutations are entering the population and causing them to become very dissimilar in their mutational profiles. But in the presence of the seed bank, you can see that there's a buffer on that molecular evolutionary change. There's still some distinctions here, but the phage effect is much more muted. So this is fresh data as well. But we have now population abundance data, phenotypic data related to the changes in the evolution of resistance profiles, and molecular evolutionary data that all suggest that the seed bank is pretty important for host-parasite interactions and co-evolution. Just three more slides maybe. And this addresses two questions. One that I've already raised: what do the phage have to say? What do the viruses have to say about this defense that dormancy can provide some benefits to phage predation. Is there any way that viruses can overcome this? I alluded to some mechanisms of entrapment in virospore formation, where it seems like the viruses can package their genome into the virospore. I'm not going to show you any data for that. The other question I have, and maybe it's not even worth bringing up, is like we do all these little experiments on petri dishes and in little flasks, and it's cute, it's nice, and it's highly controlled. But to what degree do these patterns hold in nature? Because ultimately, as a biologist, those are the things that I think I care about, is being able to know these patterns aren't just an artifact or fabrication of my imagination and what I can construct in a test tube, but that these patterns might actually be real in the natural world. And so that's not a small task to do, but one of the ways that we wanted to try to explore this is by using these virus metagenomes. So I was talking to some people at dinner last night about how biologists now have lots and lots of data available to them. We have to really generate, we've generated a lot of data here, but if you get a good an idea, there's a lot of publicly available genetic and genomic data that can be used to, ask questions about, potentially what's going on in human guts, marine sediments, surface waters in the ocean, and in soil samples. And so we know that there's about 500 genes that are involved in various aspects of spore formation. And I've talked to you that there seems to be some interactions that are going on and that there's co-evolution. So there's something called auxiliary metabolic genes. So people started sequencing viruses, and they have very streamlined genomes. We heard a little about this herpes. I think they have like 9 genes or something like this. There's not a lot of excess material stored in the genomes of viruses. What's there is packaged efficiently, and there's not a lot of junk DNA. But people started to notice that there were functional genes that were appearing in virus genomes in the ocean. So you'll recall from some of the talks earlier this week, the first thing that the hosts do when they get infected is they just shut down their metabolism. And that's not good for making virus babies. And so viruses will often contain metabolic genes that will help reboot the metabolism of their host so they can make more viruses. And in the ocean, viruses do this by stealing and using photosynthesis genes, phosphorus genes, sulfur metabolism genes, fermentation genes, methane oxidation genes, all different ways in which a microbe might make energy to fuel the production of new cells. So we started looking at some of the genomes of viruses that infect spore-forming bacteria just on a whim, and tried to blast and see whether or not there were any dormancy genes in these viruses. And there's a lot of them. And we've applied this to environmental metagenomes. So each of these genes on the x-axis corresponds to a different type of dormancy gene. And the colors just correspond to the relative abundance found in those genomes. The point is that in every single instance, every single sample that we found, there appears to be hijacked dormancy genes inside of viruses. This isn't sufficient to like wrap the story up and put a bow on it. Like you need to do more. But it's an interesting pattern. So what we did is some work. I've never thought we would be doing this type of genetic work in my laboratory. When Lior and Matthew were talking about their work, I mean, that's hardcore molecular biology, but I had a postdoc who felt pretty ambitious about this. And what he did is he found some of these sigma factors, which are involved in genes that regulate the development of the spore. And he found them in phage genomes, and he cut them out using CRISPR-Cas. And he put them back into the host and expressed them. So he increased the protein concentration of these phage-derived genes that are related to what the host would use. And what you can see in one particular instance, in this Eldridge 169, this is a gene that's homologous. It's a sigma factor that's involved in gene transcription. And you can see this is the native sigma factor F, sigma factor G, and you can see the Eldridge phage in this phylogeny is nested right within that. So it's very similar. You'd be tempted to say that it stole it from that bacteria. So we take that gene, Eldridge 169, and we put it back in the host and we express it, changes gene expression in the sporulation pathway, and it reduces sporulation by 99%. So the interpretation of those results are that those genes inside the viruses are functional. They're haven't been co-opted and used for some other purpose, like related to virus reproduction. So they maintain conservation and sequence similarity. They maintain functional capability, and when expressed, do the things that you would expect those genes to do, ultimately leading to the reduction in spore formation. And that suggests that maybe these viruses are carrying around those genes to prevent the viruses from going dormant. The logical conclusion is that if these cells here can't go dormant, that the virus would then be able to do more rounds of replication and make more virus offspring, and that would increase the reproductive fitness of the virus. So that's all I wanted to. I'm not going to give you a summary slide and belabor it. It's getting late. It's 3:30 now. You've heard a lot this week, but I wanted to try to wrap in some of the. If I went long, it's your fault because you wanted me to show those results. But these are all the pictures here of people who were involved in the research, and including people here in Germany, and people from former and current members of the lab who worked on various aspects of this work. So thank you. Good to be here.